Good afternoon, and welcome to today's webinar, Understanding Fecal Coliforms and E. coli Testing Methods for Wastewater in California. I'm your moderator, Nancy Leonard from IDEXX Water Microbiology. There will be a question and answer session at the end of the presentation, but feel free to submit your questions at any time during the webinar. You can use the Q&A tool at the bottom of your screen and type in your questions into the chat box. We'll try to answer as many of your questions as possible, but if we run out of time and you still have questions, or if you're watching this as an on-demand viewer, you may submit your questions to water@idexx.com. This webinar is being recorded, and a link with the recording will be emailed to everyone who registered, along with a copy of the slides. This webinar will introduce you to indicator bacteria, review the enzymatic test methods for wastewater quality control, QA and QC, and discuss specific wastewater regulations for the state of California. I'm happy to introduce our speakers today. Our North American Regulatory Affairs Associate, Jody Freimeyer, will be walking you through the specific wastewater regulations for the state of California. Jody received her undergraduate degree in preclinical naturopathic medicine from Johnson State College in Vermont and is currently studying at the University of Nevada to earn her master's degree in public health. She's been part of our water business for 5 years and works with a variety of regulatory agencies to help obtain regulatory approvals for IDEXX methods. She also participates in several standards organizations, including the NELAC Institute and the American Standards and ASTM. Before we get to the regulations that affect your state, please join me in welcoming Sean Dubois. Sean has a BA in biology and a BS in environmental science with a concentration in marine biology and marine ecology. Sean has been a part of our water business for 17 years, working in research and development, and in his current role, managing the product support group. Welcome, Sean.
Thank you, Nancy. So today we're going to go over a little bit about different types of methods for detecting wastewater and wastewaters for fecal coliform, E. coli, and even enterococci. But first we'll go over a little bit on indicator bacteria, as to what they are and why we look for them. When it comes to examining water for contamination, there are many different things that we'd be looking for, things that are actually very harmful. And so with the variety of things that could be out there, the difficulty of detecting them, what we want to do is find something that's more of an indicator that there has been contamination. So what we have are organisms that can be used to say whether or not there has been a contamination event. There are certain requirements for these organisms in order to be considered an indicator or indicator bacteria. First of all, they should be present when the pathogens are present. So if there has been a contamination and there are pathogens in the water, these organisms or these bacteria should also be present at the same time. When there hasn't been a contamination event, then these organisms shouldn't be present. So, and absent when not contaminated. The the indicator organisms should be in numbers that are high. Higher numbers will make it a lot easier for detection. Compared to pathogens, which can actually tend to be in lower numbers, they should be able to survive better in the water environment compared to the pathogens that would be found during contamination. If they're able to survive longer and better in the environment, then there's a better chance we'll be able to find them, and be able to find them will then let us know that there has been a contamination event. These indicator organisms should also be easy and safe to find, to grow, to analyze, as opposed to the pathogens, which could actually be dangerous to handle. So it's good to have organisms that are safe and easy to work with. Results for the testing should be fast, inexpensive, and accurate. So when it comes to pathogens, it can be usually very difficult to test for them. Tests for those kind of pathogens can actually be very expensive, and accuracy might not be as high. So having an indicator organism that is easy to grow, is safe to handle, and a test that is inexpensive and accurate is highly desirable. So one example of the perfect indicator for fecal coliforms or contamination is E. coli. E. coli is found basically in every animal, warm-blooded, birds, humans, intestinal tract. So it's there. It's not in the environment occurring on its own. It has to come from some kind of fecal contamination event. So it will not be found in the environment unless there has been some kind of fecal source. So right there knocks off a couple of the points of what makes a indicator organism. It's shed in high numbers in the feces, so it's able to be in high quantity within the contaminated waters. It's pretty resilient, so it will last in the environment a lot longer than many pathogens will. So with all of these, it kind of becomes a perfect indicator for contamination events. And once again, since it's only found in fecal material and not found naturally occurring in the environment, it is a true indicator of fecal contamination. Some other things about E. coli is it's a gram-negative, and it's thermotolerant for the most part. Most of the strains of E. coli are able to survive at elevated temperatures, so it's a pretty rugged organism to look for. That being said, when we think about fecal coliforms, usually we think they have a very defined group. First, we pull back a little bit. We look at the total coliform group. So this is a group of organisms that fall under certain criteria. They usually grow very well at body temperature, so 35 degrees Celsius. And these will include your Escherichia, so your E. coli, your Klebsiellas, Citrobacters, Enterobacters. And all of these are able to grow. Most of them are found in fecal material, but they're also found naturally occurring in the environment, once again with the exception of E. coli. A subset of this group is what has always been referred to as fecal coliforms. So these are bacteria that are believed to only occur from a fecal contamination or fecal source. And these supposedly will grow at 44.5 degrees Celsius plus or minus 0.2. But the truth is, not all bacteria that grow at this elevated temperature can come from a fecal source, and not all bacteria that are coming from a fecal source can survive at this temperature. So you can see from the nice simple diagram here is that there are some Klebsiellas or even Citrobacters, Enterobacters that are able to survive at the elevated temperature of 44.5. And there's actually a subset of E. coli that cannot survive at 44.5. So the term fecal coliform is kind of a misnomer. The better terminology is thermotolerant bacteria. They're able to survive at elevated temperatures. But for the most case, we'll be using fecal coliform because that's what people are used to when we discuss this. So when it comes to actually testing the waters for these indicator organisms, there's been a change over the years, kind of an evolution. And if we kind of look at this, we can see that at the beginning we had multiple tube fermentation, MTF. Rather a labor-intensive process. It's about 60 steps total from the start until actually running the method, all the way to confirmation. The time for the test is also relatively long. Initial steps take up to 48 hours, with confirmation steps taking up just an additional 2 days. So you're looking at almost a 4-day test in order to get your confirmed result for testing. So it doesn't exactly meet the criteria for something that's easy, accurate, and quick. So next in the evolution, you get membrane filtration. Membrane filtration definitely made an improvement on the number of steps required for testing. It only takes about 20 steps to run membrane filtration. Most results are out at 24 hours, with some tests requiring a secondary confirmation step. So an improvement over multiple tube fermentation, but still a little more labor intensive. There's a degree of skill that's required to run membrane filtration. And a lot of times there's still some confirmations that are required for testing. So improvement over MTF, but still not probably optimal. Finally, we come to enzymatic tests. So this includes Colilert, Colilert 18, and Enterolert. These tests take up to 8 total steps for quantification, with confirmed results at 18 to 24 hours, depending on the test. Much simpler to use, much more accurate, and much easier to read and run. So now I'll go a little bit over the Colilert, Colilert 18, and Enterolert products. basically going to go over how they all function. So each of these products basically has like 3 subsets of materials within each reagent. There are materials that help the target bacteria grow, gives them the best nutrient environment while not being favorable to non-target bacteria. There are suppressive qualities of the reagents that prevent non-target bacteria from growing but allow our target bacteria to grow. And then third, there is what we call our nutrient indicators, and these are what allows us to see whether or not the bacteria is present in those samples. So for Colilert and Colilert 18, both of these reagents contain 2 different nutrient indicators. One is used for the detection of total coliforms, and the other one is for E. coli. So first, for Colilert and Colilert 18, we have O-nitrophenyl beta-D galactopyranoside, or ONPG. This is used for the detection of coliforms. So coliform bacteria have the enzymatic enzyme beta-galactosidase. This allows them to utilize this nutrient indicator, or basically what they will do is they'll bind to that food source, the sugar source, the beta-D galactopyranoside, which will then cleave the bond with the O-nitrophenyl group, nitrophenyl, which will then become O-nitrophenol once it hydrolyzes. And what ends up happening is that when it starts off, it's clear, but after that bond is broken and the O-nitrophenyl is released, it will then turn yellow. So it's a color indication of the presence of coliforms. The secondary indicator in Colilert and Colilert 18 is 4-methylundifferyl beta-D glucuronide, or MUG. Basically, it's the same thing. You have a food group bound with a indicator, in this case 4-MU. E. coli has a secondary enzyme, beta-glucuronidase, that is highly unique to E. coli. And what will happen here is the same thing that happens with the ONPG, is that the enzyme will basically eat the sugar, cleave off the 4-MU group, and once that happens, the reagent will then begin to fluoresce when exposed to a UV light. So with Colilert and Colilert 18, when you run this for total coliforms and E. coli, you have 2 tests that are occurring at the same time, one for total coliforms and one for E. coli. Since E. coli is a coliform, those will have yellow inflorescence, while other coliform bacteria will just be yellow. Enterococci testing is done with Enterolert, and it's basically done with the same methodology. Once again, there's a nutrient indicator, in this case, 4-MU beta-D glucoside, with the only difference is that food group that aims more for an enzyme that's found in enterococci, and so it's glucosidase. So once again, it utilizes that indicator, cleaving off the 4-MU group, which then will fluoresce under a UV light. So running the test, once again, the whole thing is you want to have a test that's very simple and easy to run, pretty straightforward. So for all media, the first things that you want to have for quantification is, of course, the reagents, your sample, and then the quantification tools. And so for that, we have the Quantitrace system, which includes the actual Quantitrays themselves, which are the quantification device that will be used, Quantitray rubber inserts, and then the Quantitray sealer, which is actually used to seal the trays. To actually run this test is pretty simple. Like I said, up to a total of 8 steps for the entire process. So the first thing is you have your sample, 100 mLs, collected in a vessel. You add the reagent to that sample. So we'll go over Colilert and Colilert 18 first. So you'd add your reagent to the vessel, shake to dissolve. Once it's dissolved, that then gets poured into the Quantitray. Basically, at this point, the Quantitray is like a bag. And then after that point, it's samples poured into the tray, sealed using the Quantitray sealer. After that, the trays are then incubated at 35 degrees plus or minus half a degree. So once again, this is Colilert and Colilert 18 for total coliform and E.coli testing. If you're running Colilert, Colilert is a 24-hour test with a read window up to 28 hours total. Colilert 18 is an 18-hour test with results valid up to 22 hours. Both of them, once again, run at 35 degrees plus or minus half a degree. So after the trays have been in the incubator for the appropriate amount of time, they're then removed, and now what you're looking for is that indicator that for both coliforms and E. coli. So trays are looked at for yellow color development. Yellow color indicates a positive for total coliform. So you'd mark your trays with wells that have turned yellow. From there, you'll then put the tray under a UV light, a 365-degree wavelength UV light. And now what you're looking for is fluorescence. Any wells that are fluorescent and yellow indicate a positive for E. coli. And this is both the case for Colilert and Colilert 18. Colilert 18, as mentioned, can also be used for fecal coliform testing. So at this point, everything's done the same way for the test. Colilert 18 would be added to the sample, dissolved, added to the quantity tray, sealed in the sealer. And then at this point, rather than incubating at 35, the tray is actually incubated at 44.5 degrees C, plus or minus 0.2 degrees. Really want to stress the temperature, the 44.5 plus or minus 0.2. When it comes to fecal coliform testing, that temperature is very important for the test to be done correctly. If the temperature is too high, so if you're above 44.7, then the sample will be too hot, the temperature is too high, and you'll actually receive lower numbers because of the temperature basically killing off your target bacteria. If the temperature is below 44.3, then the samples are going to not get warm enough, and this will actually allow non-thermotolerant bacteria to grow, which then could increase your numbers from what they're supposed to be. So that 44.5 plus or minus 0.2 is extremely important for fecal coliform testing. When it comes to most issues that we sometimes find with customers having problems with the test, probably about 95% of them are always traced back to the temperature. And so it's very important to maintain that plus or minus 0.2. The best way to maintain that plus or minus 0.2 is using a recirculating water bath. Water baths are able to maintain temperature at a much higher accuracy than most air incubators are able to do. So for Colilert 18, once you have your trays, the trays are submerged directly into the water bath. They're not to be put into any kind of container, any kind of bag. The trays are sealed, and so no water will get into the trays and no sample will come out of the trays. So the trays go directly into the water bath. And then you want to make sure that they're weighed down in the water bath and completely submerged beneath the surface of the water. And this is important, once again, to make sure that they maintain that 44.5 degrees evenly throughout the sample. You also want to make sure that the trays are weighed down with an appropriate weight. As you can see in the picture here, we have weighted rings. Those are the best to be able to use for keeping those trays down if you need to put a weight. What you don't want to do is use something like a bottle or beaker of water, because that can actually change the thermodynamics in the water and create hot and cold spots, which would mess with the test. And you also don't put something like a brick or a rock from outside. Once again, that would mess with the temperature of the test, but you could also damage the tray. So you definitely want to use the appropriate weight in the water bath for the tray. But once again, the important thing is that you take that sealed tray and add it directly to the water bath, submerged completely beneath the surface of the water, and held down with the appropriate weight. This is Colilert 18, so the same read window applies. The test is valid from 18 to 22 hours. So after 18 to 22 hours, you remove the tray from the water bath and then count for any wells that have turned yellow. Yellow indicates positive for fecal coliforms. And once again, the reason why we're looking for only yellow and not looking for anything else is since fecal coliforms can include most E. coli, it can also include other coliform bacteria. So it's important that when reading Coli18 for fecal coliform, incubated at 44.5, that you only read for yellow color. For Colilert and Coli18, we also have a comparator available. The comparator is used when reading your results. If you have any new analyst or you're training or you're a first-time user, it's good to have this on hand. Also, you need it for most accreditations. And basically what this will, the comparator gives you is the minimal amount of yellow and fluorescence to be able to call your test positive. So most of the time you'll see a nice distinct yellow or fluorescence, but sometimes you might have wells that you might need a little extra assistance with. So taking that comparator out, you can look at it, and anything that is equal to or greater than the intensity of that comparator is considered a positive result. Switching gears to Enterolert. So once again, this is a test for enterococci. Same steps to get to the point of incubation. So once again, you add reagent to your sample, dissolve, shake, add into your quantity tray, seal, and then incubate. In the case for Enterolert, the test is incubated at 41 degrees, plus or minus half a degree. And the read window for this product is 24 hours, results valid up to 28 hours. After 24 to 28 hours, the tray is then placed under UV light and looking for any blue fluorescence. Any blue fluorescence indicates a positive for enterococci. So now you've taken time, you've incubated your samples, you have your positive results. Next step is to actually calculate the result. So to do this, you take your tray, you count the number of positive wells, and then you refer to the associated most probable number table that comes with each type of tray. So a little bit more on the trays. We have two types of trays available. A 51 well quantity tray, which will give a counting range of up to 200 MPN per 100 ml, and a quantity tray 2000, which will give you a count of over 2000 for a 100 ml sample. And this is done by the number of wells included on the tray. So the 51 well tray has 51 wells, and the quantity 2000 has two sets of wells, two sizes, 49 large and 48 small. So, for example, when counting a 51 well quantity tray, you count the number of wells that are positive. So in the case of Coliform for total coliforms, you count the number of yellow wells, and then refer to the NPN table. So an example here is if you had 4 positive wells, that would give you a most probable number of 4.2. For the quantity tray 2000, same thing. You would count the number of large positive wells and the number of small positive wells, and then use the chart for that test. So example here, if you had 48 large positive wells and 25 small positive, that would give you a most probable number of 344.1 per 100 mLs. So one of the questions that tends to come up over the years from both new and existing customers sometimes is the question of MPN versus CFU. So always cool questions like, most probable number versus colony forming unit. Is there a difference? And really simple answer is that no, there's no difference between the two. It's basically just a label or unit that's associated with a test. So in the case of a liquid-based media, such as Colilert, NPN is used. But for bacteria that are grown onto a solid media, such as an agar plate, that's when you'd use CFU, or colony forming unit. So each of them are basically just a label associated with the type of test. So if you see one, then you know the test either was done with a liquid or if it was done on a solid media. But each of them are going to be reported out as a result per 100 ml, so NPN or CFU. And really there's no difference between the two. Both of them have a 95% confidence range that's built around the result. A lot of times people feel that getting a count on a agar for CFUs is a definitive number. But the truth is there's a lot of other factors that go into running that test that actually could mean that you get lower numbers than was actually present or higher numbers. So even a plate method has a confidence range. And the same thing is true for MPN as well. Something else that comes up occasionally on our tests is whether or not dilutions need to be done when running Colilert or Colilert or Enterolert. For the most part, dilutions probably won't be 100% necessary due to the increased counting range available on the two types of coin trays. Especially with the coin tray 2000 giving an upper limit of 2,419 per 100 ml sample. But occasionally, especially when we deal with some kinds of wastewater, it might be necessary to do a dilution, or if we are collecting samples in marine environments. Some of the tests require a dilution. But dilutions are done and can be done on a sample. The important part is that the dilution should be done prior to adding the reagent to the sample. So first step is to dilute your sample in sterile diluent, and then add the appropriate reagent. And then from there, you go through the normal process of sealing and incubating. And then the important part is that after you read the result is per that dilution. And then you just need to correct for that dilution factor. So for example, if you did a one-to-one dilution where you had 50 mL of sample or 50 mL of diluent, and you got your results, you would then just take that result on the MPN table, multiply it by 2 to get back to the original answer of amount of bacteria per 100 ml. So once again, it's a pretty simple test, easy to run and highly accurate. When it comes to quality control, there's also plenty of advantages with Colilert, Colilert and Enterolert over some other methods. For example, when we compare to Colilert 18 for fecal coliform testing compared to MFC, agar plates, there's quite an advantage over the amount of QC work that needs to be done on a regular basis for the products. So for Colilert 18, it suggested that a positive and negative control, bacterial control, be done for each new lot of product received. Just to verify that is detecting what it's supposed to detect and not detecting non-target bacteria. Basically, you just do that on a per lot basis, and that's all you have to do. There's no additional QC work that's required. Whereas with the current update rule for membrane filtration for MFC, there is a lot more QC that's now involved with the product. So not only do you have to do positive and negative controls for each new lot that you run, each new batch that you make, there's also verifications that need to be done on new lots just to make sure that it's performing correctly. And of course, when you are running your set of samples, you have to do blanks, at least 2 blanks, more if you run more samples. And then on a monthly basis, you now need to confirm 10 colonies on a monthly basis with MFC. So there's a lot more QC work that's now being done with MFC as compared to Colilert 18. And it's similar to other reagents as well when you compare it to the Colilert method or Colilert 18 or Enterolert. For quality controls, it's best to get once again, a positive and negative that would be used for each one. You can either get the strains from a national collection, such as ATCC. The other way to do so would be IDEXX does offer IDEXX QC products, and we have these for all of our tests for total coliform, E.coli, fecal coliform, and enterococci testing. And basically, each of these will come with 3 vials of each organism that can be used to QC product. So, for example, if you had Colilert and then you use IDEXX QC total coliforms and E.coli, you would have E.coli, so it would give you a positive for E.coli, which would give you both a yellow and a fluorescent signal. Klebsiella varicola, which would just give you a total coliform positive, and Pseudomonas aeruginosa as a non-target, so therefore you shouldn't see any result, a negative result. but we have the same kind of tests available for both fecal coliforms and enterococci. When it comes to IDEXX products, we really strive on quality, and part of that quality system is our certificates of analysis. So these certificates of analysis, COAs, are important to have for your own records, and also for any audits where they might require you to show proof of the products that you're using. So all of these can be obtained off of our website, and there's the link here in the presentation. You can also be found by just going to the IDEXX.com/water, and then there's a link that will take you to certificates of analysis. And basically, all you will do is just type in the lot number of the product you're looking for, and then a list of products that contain that lot number will come up. You click on that, and then you'll have a PDF of the certificate of analysis that you can print off and have for your own records. Each certificate of analysis indicates all the testing that was performed on that specific lot. The way the process works on our end is that after a lot of material has been made, a sampling across that manufactured lot will go to our quality control lab. They will then run a series of tests, and once all those tests have been completed and all the forms have been filled out, that will then be reviewed by our quality assurance team. And the quality assurance QA makes sure that everything is been done correctly, all the information has been processed and is recorded, and at that point they will then release the lot for distribution. So there are several quality steps in place that we have before any product will ever leave our facility for sale. Further strengthening our commitment to quality, IDEXX as a whole has several certifications from ISO, including 9001 and 14001. And then specifically for the water QC lab, for a majority of our products, we are certified with ISO 17025, which just means that we have to go through a more rigorous examination of how we perform our tests, the testing, the amount of testing that we do, and basically the quality. So and this is done for a majority of our products. And once again, we have QC cert COAs for all of our products, whether it be our quantity trays, vessels, and all the reagents. But all of these can be obtained off of our website.
Thank you, Sean. Very informative, and we do have some questions that are coming in. So again, feel free to submit your questions so that you don't forget. If you've got questions for Sean, you can submit them right in the Q&A toolbox at the bottom of your screen. Just type them in, and when we are finished with Jody's presentation, we will then answer your questions. So with that, I now turn it over to Jody for your California-specific regulations. Jody?
Thank you, Nancy. Hello, everyone. So the U.S. EPA drives regulations in California, just like it does for all other states. Specifically, the EPA's Clean Water Act was created to control the discharge of water pollutants to surface waters, and these wastewater regulations can be found in the Code of Federal Regulations at 40 CFR Part 136. Every so often, these regulations are updated through a process called the Method Update Rule. The Method Update Rule done by the EPA in August of 2017 included the EPA's final approval for IDEXX's Colilert 18 fecal coliform method for wastewater effluent testing. After the EPA had its method update back in 2017, states that have primacy can either incorporate the federal rules into the state's rules, or the state can create rules that are equivalent to the federal rule. But if the state does create its own rule, it must be as stringent as the federal. It can't be less stringent. And just to quickly mention, there are a few hyperlinks within some of the regulatory slides. So once the presentation is available, if you'd like to review any of the regulations or contact links, you can click on the hyperlinked word underlined in blue. For example, on this slide, The hyperlink here will bring you to the EPA's Code of Federal Regulations, particularly the 40 CFR Part 136. Now, California's wastewater effluent monitoring is enforced through the National Pollutant Discharge Elimination System Permit Program, which is referenced as NPDES for short. And the California Regional Water Quality Control Board operates and manages NPDES permits. There are 9 regional water board offices or sites, which are based off of California's watershed, and the appropriate regional water board NPDES program is dependent upon where the wastewater discharge will occur. California NPDES permit requirements must adhere to the regulations outlined in the Water Quality Control Plan, specifically those that are included in the bacterial objectives, meaning the wastewater effluent bacteria requirements are dependent upon the designated use of the receiving water. So depending on the receiving water, the NPDES bacteria requirements could include testing for fecal coliforms. So stated within the individual NPDES permit are the approved analytical test methods, which usually the NPDES permit will state a general reference allowing the use of any analytical test method included at 40 CFR Part 136, which this general reference would now include the Colilert 18 fecal coliform method due to the EPA's method update rule. However, it is possible that some wastewater permits could list a specific test method instead of stating the general allowance. So if there were questions on the way a permit is written, or if the permit lists a specific test method, it's best to reach out to the permit writer listed on the individual permit. Also, I want to point out the process of changing an analytical test method for NPDES compliance is regionally specific. So for some regions, even with the permit stating a general allowance for the use of any analytical test method included at 40 CFR 136, there may or may not be additional requirements to change a test method. For example, some of the requirements might include submitting a notification to the regional board that the analytical test method is changing, or perhaps submitting a request to change an analytical test method. So to find out more information about your region, or if you have questions, you can reach out to the regional board program or the regional program manager, which here on this slide, there are 2 links, and each link will bring you to a list of contacts per region, either for the regional board program or the regional program manager. So since the EPA has adopted the method update rule, this change affects wastewater laboratories in California by allowing more analytical test methods that could be used for NPDES permit fecal coliform requirements. And this revision can be really helpful to laboratories since, as previously mentioned by Sean, Colilert 18 reduces quality control steps, reduces hands-on time, makes demonstration of capability easier for both a new analyst or for ongoing DOCs that might need to be continuously done over time, and it allows for more reliable data. So how a laboratory changes an analytical test method, in California, under the Health and Safety Code Section 100825, environmental testing laboratories that produce data for compliance purposes must be accredited through the California Environmental Laboratory Accreditation Program, which is ELAP for short. Program requirements must be met in order to add or change a new method to a laboratory scope of accreditation, meaning not only do the appropriate steps must be completed, but California ELAP will only certify for methods included in their list of fields of testing. And as of May 31st of 2019, California ELAP updated the accreditation offerings in the field of testing forms, which now complies with the EPA's 2017 method update rule. So Colilert 18 for fecal coliforms is now included in ELAP's field of testing 107 list for micromethods for wastewater. So when adding a new analytical test method, like the Colilert 18 fecal coliform, the laboratory can either request an amendment to their current certification or request a change at the time of accreditation renewal. Each process is similar, which includes completing the ELAP application, fields of testing form, and fields of testing worksheet. The laboratory will also need to include a standard operating procedure, which when writing an SOP, you'll want to include the quality control requirements, which those are listed within the Colilert 18 package insert. However, if you felt like you might need some additional help with creating an SOP, here at IDEXX we've put together an example SOP document, which is available by request. Other steps that the laboratory will need to complete are reviewing and making any necessary updates to the laboratory's quality assurance manual and passing a proficiency test. Also to quickly mention, we have put together a document that outlines today's regulatory content in the form of a method adoption guide that is specific to the state of California. This informational guide also includes the example standard operating procedure document that I just mentioned, and if anyone would like a copy of the informational guide, please reach out to us and request one. Now, I also want to briefly touch on other areas of wastewater, since in the state of California, wastewater can sometimes mean reclaimed water for reuse. So this slide is a flow map showing the various types of wastewater outputs in California. On the chart on the side matches the color of the water output, listing the possible bacteria indicators, and then shows which IDEXX product is approved and can be used for that water bacteria type. So for example, the water coming from the wastewater plant for NPDES permits, shown being dispersed to the lake, river, or ocean, is shown in blue, which if you look to the Analytical IDEXX test method approval chart, the section in blue, includes the possible bacteria indicators in the appropriate Colilert or Enterolert test method, also indicating these methods are EPA approved. Then following the land, cooling tower, industrial use of reclaimed wastewater, which this is not for NPDES permit requirements, this would be for recycled water that falls under Title 22. The chart shows the corresponding green color, bacteria, IDEXX method, and the associated approval. So today I talked a bit about California regulations, and I'd like to wrap up the regulatory portion with sharing a part of what I do, which is I follow various state rules and regulations to see how they periodically change over time. I'd like to provide a little information on how easy it is for anyone to follow and even participate in California rule changes. Anyone from the public can comment on proposed rule changes, and comments can be especially impactful to rulemakers when they come from stakeholders such as wastewater utility folks or laboratories, since you're the ones who do this type of work and may be directly affected by any rule changes. A comment submitted to the state can include things like your opinion on the rule subject, any data that you'd like to share that might be impactful pertaining to the regulation you're commenting on, or how the proposed rule change may impact your daily work. Typically, a public comment phase is open for about 30 to 60 days, but you can sign up to receive email notifications on various rulemaking activities, either at the state level or for each individual region. Then you can watch and track what's happening in your state, and the hyperlink here will bring you to a site where you can sign up if you'd like to. And that concludes what I have for the regulatory portion today. Thank you.
Great. Thank you so much, Jody. We do, as I said, we do have some questions coming in, but just as a reminder, if you go to the bottom of your screen and click on the Q&A tool at the bottom, and you can type in your questions there. We're also getting a couple of requests for the template for the SOP that Jody just mentioned. You will be receiving an email after this presentation is complete from me directly, and if you would like the SOP, you can just respond to my email and we'll make sure that you do get the SOP for the state of California. Okay, so right into the questions. Sean, the first question I have for you, is it possible to collect two samples from the same site and use Colilert 18 to become positive and Colilert 24 to become negative for E. coli?
Well, if you're collecting at different points, if you're collecting at different times, there's always a chance that E. coli could be present at one point and not for another time. So if you're collecting the sample as a split sample, same thing where depending on the concentration of E. coli, it might not evenly distribute. So you may see it come up in one product versus the other. But both products will detect E. coli if it is present in the sample. So it should be, as long as the numbers are there and it's a even distribution, if you're running a split sample, then both products should become positive for E. coli if it's present.
If it's present.
Yep.
Okay, great. Okay, Sean, another question for you. Can the trays be stacked in the water bath?
Yep.
Yep, that's it.
Nice and simple. Yeah, the important thing for, once again, when it comes to the water bath and the trays, they can be stacked, but the important part is that all the trays need to be completely submerged below the surface of the water in the water bath. And as long as they're all submerged, that should be fine. But yeah, you can stack them.
But just don't get a rock from outside.
Just no rocks.
No rocks from outside. Okay, great. Jody, question for you. Question for you, Jody. What do I do if my permit states a specific method?
Yeah, so if a NPDES permit lists a specific test method, you'll want to contact the permit writer listed on the permit, and then you can ask what needs to be done to have the method changed.
And then that contact information will be in the presentation when we send it out to everybody, correct?
You would want to contact the permit writer listed
on your permit. Okay.
Yes.
Great. Okay, thank you. And a question for you, Sean. In California, enterococci can be run by two approved methods, standard methods 9230D and also Enterolert. What are the differences between the methods? And also, when they call the method Enterolert, does that mean per the package insert?
Well, good news. SM9230D is Enterolert, actually. If you read standard methods, it calls out a fluorogenic substrate media, but it refers to Enterolert. So those two methods are actually the same. And when they call it Enterolert, what's actually being called out is the package insert. So, yep.
So yes, it is in the what the package insert says. Okay.
Yep.
Great. Thank you. Another question for you, Sean, and I believe this is referring to the slides where you're talking about dilutions. Can I add the diluent straight into the sample?
What you want to do is you can do it add to the sample, or you can add the sample to the diluent. You just want to have it in a separate container, probably from what you collected in order to make sure that you have the appropriate amount of diluent and the appropriate amount of sample to have the correct dilution. So usually, you'll have a separate container that you would put the diluent in first, and then you would add the sample to that in order to get your appropriate dilution.
Okay, great. And is it acceptable to make a dilution of the wastewater sample?
Yep. Yeah, if it's determined that the counts will be in excess of the counting range of the tray, then a dilution can be done. And when it comes to marine waters, that actually is a requirement, and it's called out in our insert for both Colilert 18 and Enterolert.
All right, great, thank you. Question for you, Jody. Does a commercial lab need to add Colilert 18 fecal coliform to their accreditation scope?
Yes. So if a sample test result is being used for any compliance testing purposes, then yes, the lab would want to add the new method to the lab scope of accreditation.
Okay, great, thank you. And then a couple of questions have come about the method adoption guide. If you just want to refer to that, how can I get a copy of it?
So we do have a informational guide that outlines the steps for adopting Colilert 18 fecal coliforms specific to the state of California. So you can email us at water@idex.com, and we can certainly get you a copy.
All right, great, thank you. And as I mentioned, I will be sending out a follow-up to the webinar that will include a PDF copy of the presentation, which will include all of the links, both the links that, all of the links that Sean had in the presentation, as well as the links that Jody had in her presentation, along with the link to the recorded version of the presentation. So you should expect to receive that within the next day or so. I'll be sending that out to everyone. And if you do need a copy of the method adoption guide, surely either send an email to water@idexx.com or just respond to the email that you'll be receiving from me. Okay, question for Sean. Is Colilert 18 more sensitive than Colilert 24?
Nope, they're equivalent. It's just the advantage with Colilert 18 is the time for result. So it's sensitive in the respect that the result will be at 18 hours as opposed to 24 hours. But other than that, they are equivalent. Just the time for result is quicker.
Okay, great. Thank you. So question for Jody. Our NPDES permit requires total coliform testing and does not mention fecal coliform. Do you know if California allows the use of Colilert 18 for total coliform testing on wastewater?
Not for total coliform for wastewater. So currently at this time, no, Colilert 18 is not approved for total coliform wastewater.
Okay, great. Thank you. Sean, question for you. Can the trays be stacked for Enterolert?
Yep. Any of our tests, the trays can be stacked. Just make sure that there's, they're not like completely filling the incubator to the point where you can't do anything inside of it. But no, they can be stacked. And there's no orientation, correct way of stacking. If you have wells up or down, that doesn't matter. But all of the products, Enterolert and Colilert Coating, can be stacked for incubation.
Okay. And another question for you, Sean. If we need total coliform only, do we need to check for fluorescence?
Nope. If you need only total coliform, then all you're looking for is yellow color development when you incubate Colilert or Colilert 18 at 35 degrees. Fluorescence is only for E.coli. Yellow and fluorescence is E.coli. But if you're just doing for total coliform, you just need to look for yellow.
Okay. And another question about fecal coliform. Can Colilert be used for fecal coliform analysis or just Colilert 18?
Only Colilert 18 can be used for fecal coliform testing. So, nope.
So no. Okay. Question for Jody. Will Colilert 18 ever be approved for sludge?
Yeah, good question. We have been getting this question quite frequently with the method update rule. Currently, the IDEXX methods were developed for testing of water matrices such as drinking water, wastewater, and beach monitoring. So at this time, we do not intend to perform a national ATP with the EPA to allow our methods to be used for sewage sludge. However, if a laboratory or utility wanted to pursue a limited use ATP for individual method approval, we could help with providing some guidance on the process.
Okay, great. Thank you. And question for you, Sean. Can I test a sample which has coloration or turbidity?
So when it comes to, yes, you can. But the
Can you elaborate?
Yeah, I can elaborate more. So depending on the amount of coloration, it may require dilution. So what you definitely don't want is if you have a sample that has, if it does have a lot of color, or especially like a lot of color that could be could be construed as being a yellow, then doing a dilution will take care of that, and then you can go ahead and run the sample that way. So you see a lot of color that could interfere with the test, then dilute the sample, and then run the test, and just remember to correct for the dilution factor.
All right, great, thank you. Is there a couple of comparator questions, but is there a separate comparator for fecal coliform interpretation?
Nope, the same comparator for Colilert and Colilert 18 that would be used for total coliform and E.coli. It's the same comparator that you would use for fecal coliform. The only difference, of course, is that you're only comparing it for the yellow color development.
Okay, and is there a comparator for Enterolert?
There is no comparator for Enterolert. What we suggest is if there's any question, if you can determine that it's blue fluorescence is present, is to run a sterile blank with Enterolert that would be sterile, and then use that. So any blue fluorescence that's greater then a negative you could call a positive. But there is no official comparator for Enterolert.
Okay. Again, for you, Sean, sorry, Jody, is it okay to use an air incubator instead of a water bath?
Assuming this is for fecal coliform, and we strongly suggest a water bath. There are some high-end or really good air incubators that are able to maintain that plus or minus 0.2. But it's best to use a water bath. So is it possible? Yes, but most incubators can't maintain that really tight temperature tolerance for the whole time for the incubation.
Okay, great. I'm just looking quickly through the questions. I think we got all of them. Some of them are repeats, so it's kind of, I'm just making sure we got them all. And we are just about out of time anyway, so if you do think of other questions after the end of the webinar, or again, if you're watching this as an on-demand viewer, please feel free to send your questions to water@idexx.com. And also, you'll be receiving a survey shortly after the end of this webinar. We'd really appreciate your feedback, as well as any suggestions for improvements, any suggestions for future content as well. We'd love to hear what you guys think. We really do appreciate your time today, and we hope that this was a good use of your time, and we know that your time is very valuable, so we do appreciate you spending the time with us this afternoon. Again, the email address is water@idexx.com if you have anything further to ask or any other feedback. So thank you so much for joining us today, and enjoy the rest of your day.